Okay, great. Well, thanks. Quick thanks to both Jen and Kyle for turning the minisymposium into a Zoomposium. So today I'll be talking about the auditory cortex, and this work is done in very close collaboration with experimentalists. So just to kind of get going in terms of the data, this is the A1 auditory primary or auditory cortex. And usually what is done here is that you play a series of pure tones and you see what the neurons are most tuned to. And it turns out that they do usually have a best frequency. And you can see that kind of color coded here. And again, this is the auditory cortex here. We're going to be focusing on A1 specifically because it really has this nice feature that everything is pretty much on this tonotopic map. And we have this very strong correlation between space and feature preference, right? So in this area, we have neurons that prefer low frequency sounds. And as we move over to the right, it increases. And over here, we have neurons that prefer high frequency sounds, right? So this is just kind of these experiments in which they play these pure tones. But of course, mice do not just have these pure tones. And these are now recordings of mouse vocalizations, and we see that there are actually, frequency sweeps. And it's much more complex than just a fixed tone. And we see that some of these sweeps go from high frequency to low frequency, and others are in the upward direction. So if we go from low to high, it might sound something like this. And then from high to low, kind of goes down like that. And we see, in terms of these mouse vocalizations, we're all over the spectrum. So now if we collect this data by different types, whether or not it's kind of like just a short sound, a more long courtship sound, or these long kind of stress sounds, and we see what kind of frequency sweeps, how fast are they? Are they in the upward or downward directions? We see distributions that look normal in nature, that are kind of almost centered at zero, and they really span like a wide range of sweep speeds, both in the upward and downward direction. So it's very important for these neurons in A1 to not only detect pure tones, but to be able to detect frequency sweeps. And that's exactly what we see. So now we go back and we're recording from neurons in A1. But now instead of playing a pure tone, we're playing these frequency sweeps. And we see cell 1 has this interesting feature that if you play a downward sweep, basically there is no response. However, you have an upward sweep at different speeds, we see the cell does respond. Cell 2 has kind of the opposite effect. Cell 2 seems to prefer downward sweeps and has very little response for any upward sweep. Cell 3, on the other hand, responds to both upward and downward, maybe a slight preference here for the upward direction in this peak. So this leads to this this index, the direction selectivity index, in which we can characterize how strongly a neuron prefers either the upward or downward. And that's just kind of the integral of the upward direction minus downward divided by the integral of upward plus downward. And when we see that it's actually, in terms of the ability of a neuron to respond to either upward or downward at various speeds, speed doesn't really come into play. A neuron is very good at preferring either kind of the upward or downward direction, kind of across these different sweep speeds. And now if we go back to the tonotopic map, we see another very interesting feature that pops out of the data. And that is, if we're in this low preferred, like this is again, pure tone, a pure low frequency turn, we tend to have a positive DSI. That means that those neurons on that side of the axis prefer a upward sweep. As you move across and you now go into the higher frequency sweep area, you now are getting to neurons with a negative DSI, meaning that they prefer a downward sweep. Now, kind of the leading hypothesis in this area is the fact that, this DSI, this direction selectivity, arises in A1, meaning it's not inherited. You can imagine that it's, when a tone is played, it's this region of the cortex is receiving feed-forward input but this direction selectivity is arising through recurrent dynamics in this region. So that's one of the questions that we kind of want to answer. How is that possible? How does this direction selectivity arise via recurrent connections? We can kind of take a closer look. So this is now whole cell recording from an upward preferring neuron. We're fixing voltage is -70, and we're recording, the kind of excitatory currents into the cell. And what we see is, again, this is an upward preferring neuron, and what we see is that if we kind of take a close look here, is that we have these very fast excitatory responses that arise in the upward direction. Another feature that we notice is that, indicated by these stars, are network suppression that follows this fast excitatory input here in the upward direction. But this network suppression is also noticeable in the null direction or the non-preferred direction. So what does this mean? If we take a zoom in look, so we look over here, we see in the preferred direction, these neurons are able to have a fast excitatory response, which is blocked in the null direction. We can fix the cell now at a positive 20 millivolts and look at inhibitory currents. And we actually see a similar story, is that in the preferred direction, we have this fast inhibitory response followed by network suppression. And network suppression is also prevalent in the null direction. Again, so we see similar story. In the preferred direction, we have a fast inhibitory response that is blocked in the null direction. And this leads to this observation that in the preferred direction, we kind of have this amplification of charge in both the excitatory and inhibitory component. All right, so this leads to a very interesting modeling question. Because when you think about this model of A1 or the cortex in general, you might think that you want to start with two populations, excitatory population, inhibitory population, both having kind of these recurrent connections and then talking across the populations, with both of these populations receiving feed-forward input. And maybe you could look at kind of a mean field model of the rates of these populations. Well, why would you start with that? Well, there's plenty of examples in the literature that does a lot with these EI networks, whether it's the visual cortex and orientation tuning, odor representation, or even some work in the auditory cortex. These EI networks have been shown to really capture a lot of the experimental data. However, here we have a problem because the model suggests that both the excitatory and the inhibitory components are suppressed in the null direction. So that means that this kind of standard EI network will not get the job done. We need another player to come in. Luckily, there are other players that we can choose, and that comes from the fact that these inhibitory neurons, there's not just one type. There's actually subtypes of inhibitory neurons. PV and SOM are two of the main types, and the two types that I'll be focusing on for this talk. They make up 70% of all neurons. These are just genetic markers that can be used to identify these subtypes. But beyond just these markers, they vary in many ways, whether or not they're able to have spike frequency adaptation. Their synaptic properties vary, whether or not they're kind of depressing connections, as well as how they're connected in this network. And that's what I'll be focusing on here, just to kind of illustrate kind of the differences between these subclasses. So here, you can kind of view this as kind of the effective connectivity matrix. Basically, it says how strong are these two populations connected. And we have pre, so it's going from PV to E, PV to PV, and then PV to SOM. So that's how you can kind of read this table. And we see that a couple of these are actually much smaller than the others. So the first step I'm going to be is basically just apply a threshold and basically take those terms, because they're just significantly weaker than everything else in this matrix, I'm just going to take them to be 0. And now going row by row, we see this first row can be read that PV has a strong connection to E and a strong connection to itself. SOM neurons, though, vary in the sense that they don't have this recurrent connection. Instead, they have the strong outgoing connection to excitatory cells and a strong outgoing connection to PV cells. All right, so now we're going to be focusing on this network, but of course the excitatory neurons also have connections, and so we'll add those in there. Another thing that is interesting to note in this network is if you talk about this feed-forward component, we only have feed-forward components going into the excitatory cells as well as the PV cells. The SOM cells aren't actually receiving this feed-forward component. So this is kind of the network, and it really shows that PV and SOM cells are really kind of in the network differently, and they're primed to kind of do their own, have their own roles in this network. If we look at it a little closer, we see that you know, the standard EI connection exists in this network if we just look at E and PV. But we really don't want to look at that. We know from the data that we're going to have to look at this full system here. Experimentally, like I said, we can identify these cells, and our collaborators have gone in and silenced PV cells and asked, what does that do to direction selectivity? And we see here, the black is the control, the orange is when PV cells are silent, or at least blocked. And we see that not much of a change occurs. And DSI is pretty much left untouched. We can do the same thing for SOM cells. And we see that in the preferred direction, not much changes. But in the null direction, when you would imagine a weaker response, when SOM cells are blocked, they respond, these neurons respond strongly. And that basically means that this neuron that we're recording from has lost its ability to be selective to a specific direction. And that's seen here. We're below this one-to-one line, meaning that DSI has been very much weakened in this case of SOM blocked. All right, so just to kind of recap, there's a lot of experimental data, and these are the features that we want to capture in the model, is that A1, we have, you know, this observance that cells are selective to a certain direction of frequency sweeps. And depending on where you're on the axis depends on whether or not you are an upward preferring neuron or low preferring or downward preferring neuron. There's this amplification in charged in the preferred case versus the null case. And the SOM inhibitory neurons really seem to be the key player in this network. Okay, so now we're going to shift. Those are what we want to capture with the model. Now we're going to shift and actually talk about how we construct the model. So we're going to have a spatial model here along the tonotopic axis. So we have this nice feature of A1 in which feature and space are really strongly correlated. So we're going to have this kind of spatial component. We're going to use a mean field approximation, basically just focus on rates. How do the rates of the population change as you move across this axis? Another key difference between PV and SOM is how fast they're recruited. SOM cells are recruited on a much slower time scale. So we have this added equation here that basically passes through the rates through another temporal filter to represent that another significant difference between inhibitory populations. As a result, because we have E, PV, and SOM subtypes, we're going to have 6 total equations to be dealing with here. And lastly, I want to highlight this B term here. This is the input. This is the feed-forward input, which we're going to say is the same regardless if it's an upward or downward sweep, right? The direction selectivity is not inherited downstream. So it's just going to be kind of this Gaussian that kind of sweeps across the space. And it's only input to E and PV cells. SOM cells do not hear this input. Okay, so what's happening? What's the connectivity along this axis? Well, we can look at a specific point, and this is exactly what we saw earlier in the talk in terms of the connectivity. And this is just kind of the local. But there's very interesting work that's known in terms of how connections work spatially. So we can see from E to PV, we have this kind of moderate spatial component in terms of, you know, what cells are talking to what E cells. So this is that K term in that last equation, which is a kind of a Gaussian. And the big difference here to note is that if you look at the projections into SOM and projecting out of SOM, it is much broader. Basically, we have a much broader range of SOM projecting and receiving input from. And this goes not just from E connections, there's no PV to SOM, but from SOM to PV, again, this very broad connection. And we think that's a very key feature of this model. Again, I mentioned that this is a rate-based model. One thing that's not clear in this equation is that we're actually also imposing a threshold nonlinearity here, in the sense that in this cortex, you can imagine that theta alpha is kind of the steady state firing rates, and it's relatively low in the cortex. So that means that if the input is strong enough in the negative direction, we imagine that these cells can actually be silent. And that means that the rate goes to 0. So we don't let it go into a negative rate. We have it thresholding at 0. And we see with this model, with the components that I just mentioned, we do in fact have DSI. We define DSI in a very similar rate. We have the integral in the upward direction minus downward, divided by upward plus downward. And we see this is now from the model. And as we compare to the experimental data, we see these key qualitative features arise in terms of low frequency neurons preferring upward sweeps, and then up here it's downward sweeps. So now I'm going to play this movie that kind of shows what's going to happen. I'm going to play this movie twice to really kind of go through the details of why this model does what it does. So we're going to be looking at two neurons here. One is a low frequency preferring neuron, and one is upper. And here you're going to see an equation for rates, as well as the inhibitory inputs, as well as the feed-forward input. So as I play this, we see that we have the feed-forward input that's not changing, right? It's just this Gaussian that's it's this traveling wave across the tonotopic axis. We see that we have this period of silence in both of these types, and then here we're going to have kind of an upward response. As the sound gets to this area in the cortex, we have a response that then goes away. All right, so that's a lot to take in. So now I'm going to play it again and kind of go through exactly what's happening in this model. So if we pause it right at the beginning here, we note that as soon as the sound arrives, notice that the system's just at steady state. There's no, all the currents are just kind of at steady state. There's no strong inhibition. As a result, this cell at this lower part of the tonotopic axis is able to respond very strongly. It just has the excitatory feed-forward input that's really driving it at the start. Over time, we see that the PV cells also start to turn on, but these SOM cells, which have the slower recruitment timescale, starts to turn on, and they end up, because of their broad projections, end up projecting ahead of the feed-forward input. Oops. And as we move the movie forward a little bit, we notice that the SOM cells are so strong that leading the feed-forward input is this silencing, right? The inhibition is so strong that it's completely silencing the excitatory cells. So we have this leading suppression for this upper preferring neuron. We have a trailing suppression here, but that doesn't really come into play because the cells already responded very strongly. So by the time that the sound gets to this high frequency neuron, the 60 kilohertz neuron, it has to overcome the suppression that's already there before responding. And the end result is a much lower kind of peak. So again, this just illustrates kind of all these details that I just kind of talked through. Again, it's very important to note that the feed-forward input is the same whether it's upward or downward, so it's not inherited. We have total excitatory current, PV current, and SOM current. The SOM, we really see these broad projections coming into play. And as we look at the firing rates, we see again this leading suppression that happens in the blue curve, it's trailing suppression for the red curve. And we see that we do kind of capture this amplification of excitatory currents that's seen in the experimental data. Similar to the experiments, we can now go in and block SOM cells in the model. We do this by having an inhibitory current added into the model, as well as just weakening their overall effective conductivity. And we see that when we do that, we do effectively eliminate DSI, very similar to what was seen experimentally. And part of the reason, if we look at the blue curve here, we don't have this leading suppression. The cell never goes completely silent. It's able to respond much stronger than it was if it had been silent. There's a difference in peak, but if you look at the integral of these two curves, they're actually very similar. That's seen here, kind of the difference between, we don't have this amplification of total excitatory charge that we saw previously. What happens if we block PV cells? Well, similar to the experimental data, we see that it doesn't have much of an effect on the DSI. These cells are able to respond selectively to the different directions. And we see, this leading suppression is very much, present, if not perhaps a little stronger in this case. So now a thing that we can do that, experimentalists can't do is we can now kind of ask, okay, what makes some cells kind of primed to do this? Why are they kind of the key component in this direction selectivity? Well, one thing that we can do is we can change their spatial scale. We can say, okay, what happens if we basically make them on the same order as PV and on the same order of PV cells? And we see when that happens, we eliminate DSI. Again, this leading suppression is not seen here in this firing rate, and we have no amplification here. So the spatial component is very much key as to why some cells play such an important role. You might also ask, okay, what about the thresholding I mentioned? Is that a kind of a key component? Or you can imagine that what happens if we shift up the baseline of the firing rates so that this nonlinearity doesn't actually come into effect. If that's the case, if we're up here, we can simply linearize the model now, rerun kind of the simulations, and we see that DSI is completely eliminated. And it actually can be shown that this is always the case. If you have a linear model, DSI is impossible. And the proof it's kind of a cute little proof. It's just a couple lines here. We have kind of this equation here. To compute DSI, we just take the integral of both sides. The fundamental theorem of calculus is always handy. Comes through and sets the left-hand side to be 0. The right-hand side we can simplify, kind of pass through the integral through. W is not changing, it's the connectivity matrix. As a result, we can calculate the change in rates. We note that again, B, the feed-forward input, is the same for both upward and downward sweeps. If that's the case, then the change in rate is the same in upward and downward, and DSI will always be exactly equal to 0. So in this case, we've shown very clearly that this linear regime does not work for this model. This is a very key component of this model to get what we observed in terms of DSI. So then just to kind of wrap up, what are the key components that we learned from the model that helps explain the experimental results? Well, it's really this broad SIAM inhibition that leads to this network suppression. As well as the fact that we are kind of in this non-linearity of this threshold, the fact that these cells go silent when they are input with a heavy and strong inhibition. That's also a very key component to get direction selectivity. And then just to kind of connect kind of to the larger picture as a whole, I think it's very important to note that inhibitory cells are not just I, it's not just EI, it's actually E plus many different subtypes that vary. And experimentalists know this. Experimentalists can now go in and silence specific subtypes of cells. So I think it's important for modelers to be able to kind of keep up with that and be able to incorporate these more complex subtypes in. Because it turns out in this case, SIAM cells, this specific subclass was really key to explaining the experimental data. All right, with that, I'll just note that a lot of this work is coming out hopefully soon. It's currently under review. We're currently in response to some comments. And then I just want to thank everyone in the Neuron group, of course, Brent, as well as our experimental collaborators. So with that, thanks.
Brilliant. Thank you very much. That's a great talk. Brent's looking a lot beardier than the last time I saw him.
Yes, he has grown that beard.
Okay, brilliant. Are there any questions for Greg?
Yeah, I have one. Can you hear me?
Yep.
Oh yeah. So the SOM inhibition, is that intrinsic? Is that some GABA B component, or is it like some network effect that's slower? I mean, the time scale, I guess, of the inhibitory synapses.
Yeah, no, that's a good question. I believe it is GABA.
Okay.
Yeah.
I mean, I guess I have a question as well. So in these experiments and the model, you're using these pure tones, and of course in the mouse and humans, of course, when we're going around, we're seeing a combination of all sorts of things. So how do you think the inhibition that you're getting from the SOM, given that it's quite broad, works with having lots of different sequences of tones that both go up and downwards sequentially?
Yeah. So, I mean, how does that, when you start, so far, this was kind of a clean experiment in the sense that we had, just this frequency sweep up and then down. If you have more of one of these complex kind of sounds. And I think, I don't know, that's a difficult question. I think it's one of those things that because SOM cells is this broad, but they're also kind of lagged. I think it would be interesting if you kind of have those kind of interplay things. And right now, I don't know, it's kind of unclear how that would all fit together.
And one very, very quick question then. Just thinking back to the experiments you did in the in silico experiments you did at the end, what role do you think that the slower buildup of activity in the SOM plays in the results that you talked about?
Yeah, I think the slow buildup actually did play a really key role in terms of having, if you look at, if I just go back, let's see. Having this broad DSI, the fact that we have positive DSI kind of in this half plane really comes from the fact that it's this kind of this slow SOM buildup. If it's fast, I think you would imagine this will drop much quicker. So we still have a peak here, but if SOM cells able to respond very quickly, this would drop and we'd almost get kind of a very kind of sharp step function.